You're most welcome to this talk. Now, today we're going to be looking at research from Germany that shows that between 86 and 90% of COVID tests using the PCR, polymerase chain reaction methods, were false positives. In other words, COVID was massively overdiagnosed. 86% were false positives. That means 14% were accurate diagnoses of COVID. It was massively overdiagnosed by this PCR test. And this is the paper that I'm going to be giving evidence for that here. It's actually quite an intelligible paper. You can see it there. It's all in the public domain for yourself. Now, just think about the implications of this. It looked like there was 7 or 8 times more COVID than there actually was, causing infections as judged by this PCR test. And I've got a few notes here actually. I'll just show you them very briefly of how far out things were. Now, this was the Lancet, very prestigious medical journal. Not making it up, check it out for yourself there. So that is the Lancet, the information that gave. And what that actually said, the Lancet said the specificity is greater than 95%. In other words, this is saying that 5% were, at most, 5% were false positives, whereas this current data is showing that 86% to 90% are false positives. A bit of a difference. Given that these figures are probably correct, how did the Lancet get it so wrong is a question. The UK government, let me show you their site there. This is the UK government site where they talk about impact of false positives. And what are they saying? Well, the figures that they gave, this is 2020. The UK government are saying, there we go, 2.3% of false positives. So the Lancet at most 5%, not 86 to 90. So the UK government figure was 2.3% false positives. The actual figure seems to be 86%. And the hallowed New England Journal of Medicine, again, this is the reference here. Don't take my word for it. They said, what did they say? 1.7% were false positives. So if this German study is right, if this study is correct, this one, and it's looking pretty good. I'm going to show you the details of it because I'm convinced by it. That's why I'm telling you about it. That means the Lancet was hopelessly wrong. Hopelessly wrong. That means the British government was hopelessly wrong. And that means that the New England Journal of Medicine was hopelessly wrong if this German study is accurate. So let's have a look at it and see what we think. So here's the study here, a calibration of a nucleic acid polymerase chain reaction by antibody IgG tests in Germany. So if you have an infection in the blood, that is an antigen. The SARS coronavirus 2 is an antigen. And that will cause the body to make antibodies by definition. So the virus, unless it kills you first, will cause you to make antibodies. First you make one called immunoglobulin type M. That's immediate, very quickly. Then over the course of days to a week to up to 2 weeks, sometimes a bit longer, depends, you make another one called IgG, immunoglobulin type G. So they were able to work out who had a positive PCR test and who actually later on tested positive for the immunoglobulin type G, indicating that they actually did have the virus in their blood causing an antibody response. And that's how that worked, and that's pretty good science. I don't really see how you argue against that. Okay, you've got a PCR test, might or might not be right, but if you're producing IgM type, immunoglobulin type G, immunoglobulin type G, then that means you definitely had the infection. So they know who definitely had the infection, and it didn't work out very well. 86% were false positives in the time frame. Of course, they took full account of time frame. Now, this paper is quite involved and complicated. In principle, it's simple, but they're able to adjust for the time frames using all sorts of mathematics. Now, I don't pretend to understand this, of course not, but it is peer reviewed, so I take their conclusion as being accurate. So what they did was they looked at the PCR tests, and then at an appropriate time later they looked at the antibody tests and found that most of the people who tested positive for the PCR test did not go on to seroconvert within the proper time frame. In other words, they were showing false positives. And as a result of all these false positive tests, we had all these huge changes in society. So, German authority accredited laboratories. So this is proper accredited laboratories in Germany. This is not some bloke working in his garage. And this covers 90% of the SARS coronavirus 2 preliminary chain reaction tests done in Germany. So again, this is not a small sample. This is 90% of the PCRs that were done in Germany. You can see statistically that's going to be incredibly good quality data. March 2020 up until January 2023. And so not only did they look for the PCR, and they conducted serological mass tests for immunoglobulin type G antibodies. If the immunoglobulin is there, they had an infection. If the immunoglobulin is not there, within 2 or 3 weeks after the PCR test, they didn't have the infection. This is proper hard science that shows that the Lancet, the British government and the New English Journal of Medicine, if this is correct, it shows they were wrong. How could people make a mistake? How could the British government, for example, make a mistake of such magnitude? How could they get it so wrong? And yet I'm giving evidence on this video that they did. Are they grossly incompetent? How did they get it so wrong? Observed IgG seroprevalence trajectory. In other words, after that, did they convert to IgG, immunoglobulin type G? All this immunoglobulin type G it's kind of a Y-shaped molecule. It's like this, really. It's got, that's basically it. I mean, it's a very complicated molecule, but it's got a bit called the multivariable domain on the end, like that. That's a particular shape. And it's that shape that fits onto a particular viral particle. So let's say the spike protein. It fits onto the spike protein like that. So that would be the virus. And then that there could actually stick onto another virus there like that. So what it's done, it's now clumped 2 viral particles together. Then of course you could have another antibody here, another one there, like that, another immunoglobulin type G there, and that could fit onto that spike protein there, and then that could fit onto that one there, and that would be another virus there. And I think you can now see we've clumped together 3 viruses. We've got agglutination, and once they're all clumped together, they can be eaten by phagocytes. There's other ways that antibodies work, but that's one of the ways. So these immunoglobulin type Gs, Ig for immunoglobulin type G is this particular Y-shaped type, vital for immunity. And if there's infection, you will get the production of these immunoglobulins. Right, therefore, test positive fraction of PCR and immunoglobulin tests could be correlated. A value of 0.14 was found fitted the scaling parameter. So basically what they're saying is 0.14, which is 14%. 14% of people that tested positive for the PCR went on to develop the immunoglobulin type Gs in the correct time frame, the 2 or 3 weeks after the positive PCR test. 14% did. That means 86% didn't. Couldn't make this up. They were so wrong. So only 14% of those who were tested for PCR positivity actually became infected with SARS coronavirus 2. Because if they didn't become infected with SARS coronavirus 2, they would not produce the immunoglobulin type G to SARS coronavirus 2. Now, also interesting to know, a quarter of the German population carried immunoglobulin type G antibodies from natural infection in their blood at the turn of the year from 2020 to 2021. In other words, before vaccination started. Before the first vaccine was given in Germany, 25% of the population already had natural immunity. They already had natural immunity, which is way better than vaccine-induced immunity because it's polyclonal, it acts against lots of different parts of the virus, and it's longer lasting. So by the time the vaccines came, already 25% had immunoglobulin type G, and of course the immunoglobulin type G means they actually had the infection. Now, they also did a second independent analysis. This is one of the things I like about this paper. So it wasn't just one. So it wasn't just one methodology that they used from these labs. They also got data from the Robert Koch Institute, which is like the definitive thing in Germany. And what they found there is that the specificity ratio was actually 1 to 10. So what they found there was only 10% of people who tested positive for PCR went on to develop immunoglobulin type Gs. In other words, 90% didn't. In other words, it had a 90% false positive rate. For every 100 people being tested for SARS coronavirus 2, 90% showed false positives. They weren't actually infected. So that was a 1 to 10 one, so that was even more convincing. I'm not going to go into the details of that second method, but they had two methods to triangulate that. Aligns well to perfectly. It's a very good correlation with the IgG positive fraction 92% reported by the Robert Koch. So the Robert Koch data even more convincing than these independent labs. So that's basically what I want to say on this video. The paper's there, check it out for yourself. 90% of these polymerase chain reactions. So the PCR test is not these silly little plastic lateral flow rapid antigen test things. It's when you take a swab and it goes to the lab. And they multiply it and multiply it and multiply it so they can find the genetic material from the virus. 90% of those were showing false positives. And if it's 86% to 90% were showing false positives, indicating there was 86 to 90% more infections than there actually were. Like getting on for 9 times more infections than they actually were. That's what I want to say. I'm going to give a bit more detail now for those that are interested. That does help to clarify if you want to stick around. So breaching the epithelial mucosal barrier, breach, meaning the invasion of a person's organism by the actual viral material. So what he's saying is a lot of the time there was not breaching of the epithelial mucosal barrier. In other words, the mucus lining. So if we have the lungs here and the major bronchial passages branching out into smaller and smaller bronchioles. This upside down tree that is the amazing fractal pattern of the lungs. This of course is lined with mucus. Most of the time that mucus was attracting the viruses. They were sticking to that and not getting into the body to infect the blood. It is common scientific terminology to say that the person became infected, not when there were some viral particles in their respiratory tract. That's going to be all the time, really. But when active viral material enters the mucosa or epithelial lining, may be bound and possibly already neutralized by immunoglobulin type A. So So what they're saying is you're not infected when you get the odd viral particle here in the respiratory tract. That's going to happen because the virus is everywhere. You're only positive when you get the virus in here in your blood. And it's only when you've got the virus in here in your blood that it's going to stimulate the development of the immunoglobulin type Gs, indicating an actual genuine infection. So it's common to say in scientific parlance that someone's infected when the virus is in the body's organism, inside the blood, the body fluids, stimulating. So it's the antigen that stimulates the production of the antibody. That's what infection actually means. If there's a lot of COVID around, obviously there's going to be viral fragments in someone's respiratory tract. But we'll see it's even worse than that, if you just give me a minute. In most infection cases, particularly when symptoms occur, immunoglobulin antibodies will also become detectable in the blood. So if someone becomes symptomatic, they're probably going to develop immunoglobulins. The presence of immunoglobulin G in antibodies in the blood is representative of the body's immunological memory of infection. In other words, there has been an infection, and the body's very clever. When there is an infection, it remembers it, so it can combat it better in the next time. It remembers that particular antigen, and it can mount an immune response much quicker and much better because it generates memory cells. Also indicating that IgG has been produced. IgG antibodies remain detectable for up to a year in at least 90% of naturally infected SARS coronavirus 2 positive individuals. So whereas the vaccine immunity was only lasting for a few weeks to a few months, the immunoglobulin antibodies remain detectable for up to a year in 90% of naturally infected individuals. So during the pandemic, natural immunity was poo-pooed. People said vaccine immunity is better. It's not. Natural immunity is way better. And then they said, well, you better have hybrid immunity. You better have the vaccine and the natural infection. No, the natural infection has antibodies that stick around for over a year. And even then, after that time, there'll still be memory lymphocytes that are there for decades, not just a year. So natural immunity was good, but it was poo-pooed. PCR tests merely detect the presence of fragments of viral genetic material, not necessarily active infection. So if a couple of viruses have got into the respiratory tract, it might have been broken down by the immunoglobulin type A. That's the one that's in the mucus. Then you might have some fragments, but those fragments wouldn't have been viable. They wouldn't have been able to reproduce. They couldn't cause an infection, but they would give a positive PCR test. So there could have been some viral fragments there. The relationship between PCR and immunoglobulin test results is crucial, since PCR positive counts were widely interpreted as proxies for actual infections. So PCR tests were counted basically as infection numbers. That was wrong, and served as the basis for public health policies. So public health policies were instigated based on a times 7 to times 8, even to times 9 or 10 overestimation of the amount of infection. Individuals whose PCR test requires CT, that's replication values above 30, are commonly not to be considered infectious. Many tests were conducted at CT values of over 40. So if you only get a positive PCR test after 30 cycles of doubling the amount of genetic material there, that shouldn't be counted. They shouldn't be counted as infectious. But then they went on doubling it another 10 times and still counting in that as positive. Why did people not realise that CT values of 30 indicate that there's no infection? So CT values of 40, which is multiplying it hundreds, thousands of times more, are even less significant. It becomes meaningless. And this was one of the most frightening things, given that CTs are often done at 30 replications. PCR analysis produced positive results on water controls at cycle thresholds between 36 and 38. So in other words, if you put some water into the PCR test and put it through 36 and 38 cycles, it would become positive. Water, pure water, would become positive. It would show a positive. And yet they were being done at 40 when the infection threshold was 30. We could go on longer, but in short, a PCR test provides a snapshot of an individual's current exposure to viral genetic material at the outermost layers of the body, not infection. So that is really quite appalling that 86% to 90% of PCR tests were false positives. The world was turned upside down based on those false positives. And that's inconsistent with the Lancet, which said it was only 5% or less. The UK government, which said it was 2.3%, not 86%. And the New England Journal of Medicine, even in 2024, that said it was 1.7%, not 86%. How did they get it so wrong? No one's saying it's deliberate. But how did they make mistakes of such magnitude? Is an open question. We'll leave that there. Let's hope PCR tests are used with more prudence in future. But just think of the implications of overestimating the amount of infections by 7, 8 or 9 or 10 times. And all the personal and economic distress that could have been prevented. At the time, when we were looking at this at the time, these were the figures that we had. If it was in the Lancet, that was like written in blood, it was like definitive. 2020 UK government website, what other information did we have? How could we give informed consent based on such erroneous information? Well, we didn't, did we? But for now, thank you for watching.